is here.
Yeah, I mean, we also have a lot of students of Professor Coutinho and maybe your colleagues as well.
Yes, yes, I can see them.
Okay, so here we go. A very good evening to one and all. I am Sakshi Yogendra Kasat, President of American Chemical Society, Bombay College of Pharmacy Student Chapter. And on behalf of the entire chapter, I welcome all of you and of course our guest speaker, Dr. Alpesh Malde, who has joined us from Australia. And he's also a BCP alumnus. And we are really, really glad to have you here, sir. So this is our monthly webinar for June and our very first research talk where we are going to learn and understand how a drug binds to its target at an atomic level, just as the title says. And well, the question, how does the drug act at an atomic level, seems quite simple, but to reach the answer, the path is not that straightforward, of course. So what all efforts are taken, computational studies, simulations, experimentations, what is the chemistry and how it is explored. So let us learn it all from our expert today, Dr. Alpesh Malde. I once again welcome you all. But before switching right into the knowledge and learning mode, let's get to know our speaker. And so here I invite Saurabh Kundekar. He's the communication team member to introduce to us our guest speaker of today. So please, Saurabh.
Yes. Yes, thank you, Sakshi. We are really grateful to have Dr. Alpesh Malde at our chapter today. Dr. Alpesh Malde pursued his bachelor's degree from Bombay College of Pharmacy, Mumbai University, and Masters of Science degree in Medicinal Chemistry from National Institute of Pharmaceutical Education and Research, Naipur, Mohali in 2001. He completed his PhD in Pharmaceutical Chemistry from Mumbai University in 2007 and later pursued postdoctoral studies and further research fellowship in molecular dynamics simulation and free energy calculation till 2018 at University of Queensland, Australia. He has also been the co-founder and developer of the automated topology builder and respiratory. Dr. Alpesh Malde is currently working as a research fellow at the University of Glycomics, Griffith University, Gold Coast, Australia, and leads the computational chemistry theme, focusing on the development and validation of computational method, force field parameter, and their application in structure refinement and drug design. His research area encompasses development and validation of computational method using computational approaches including molecular dynamic simulation, quantum mechanical, and free energy calculation to reconcile, complement, and guide experiment. He has received various awards and honors, some of which are University of Queensland Research Award, Australian Research Council APD Fellowship, University of Queensland ECR Award. Now I invite Dr. Alpesh Malde to please begin the session. We also have question and answer session at the end. So please, if you have any questions, put your queries in the chat box or unmute yourself to us. Over to you, Dr. Alpesh Malde.
Thank you, everyone, especially Sakshi and the whole team at ACS and Elvis also. With the last few discussions that I was told that I'll be speaking at some stage at this forum, and which is nice. And I'm going to keep this talk a little bit casual, and in between I'm going to add few things. And again, as it was said, I did my B.Pharm from Bombay College of Pharmacy, and I got, of course, a special attachment to that college, that place. And of course, I came back to do my PhD, and it was an interesting time. So I did my M.Pharm or what they call MS Pharm from Naipur, and I continue to do my research at Naipur for some time. Then I wanted to come back to Bombay. Came to college, met sir. Sir said, "Come back." I said, "Okay, let's do it." So during my PhD, I spent half of my time at the Bombay College Pharmacy doing computational work with sir, and half of my time I spent at TIFR doing NMR of small peptides. So that was my second experience at doing something in the lab. So while I was doing my master's at Naipur, most of the work on my thesis was on the computational work. But the end of that thesis, I spent some time in the chemistry lab doing some organic synthesis. So those part and NMR are the only things I have done in the wet lab. but out of that, most of my work has been computational and method development. So what I'm going to do is now jump onto my talk and see if you all can look at the slides. So the most of my work has been based on answering two basic questions: that how a drug molecule binds to a protein. What we need to understand is the two aspects. Uh, so just try to, okay, I can't get the pointer here. Sorry. Okay, never mind. So use computational methods to understand how drugs bind to the protein. So we generate a lot of data from experiments using, say, X-ray crystallography, NMR, mass spec, or glycan array. When we generate all this data, what they are giving us are some signals or some information. We can't see exactly what's happening with the structures. So what happens? We combine those experimental data with some models, structural, and the description of this model, and to build that this is how it's likely to look like an atomic level. And then we can use that information to design, say, drug molecules or the biologicals. So most of the work I have done has a basic theme of either to interpret and reconcile the experimental data, or use that to complement and guide experiment. For example, that we look at some of these drugs binds to protein, and then we say how do we optimize the binding of those drug molecules, and then we design and test computationally before we go to the lab. And some of the cases replace the experiments. In this case, like, it's not that practical or possible to, say, screen hundreds of thousands of compounds in the lab. So what you do is computationally screen them and shortlist the most probable ones which are likely to work and go back to the lab. So this is the overall theme of the work. And to begin any of the structural work, the major source of the structures is the structures we get from the experimental method we call X-ray crystallography, which has become an indispensable tool in structural biology. Now, what happens in the X-ray crystallography? On the right-hand side of the slide, you can see there's a crystal. You get the diffraction pattern, and then you process the data, and you get the electron density map. Now,
Dr. Alpesh?
Yep.
Sorry to interrupt. Could you please try changing the slides? We are not able to.
So, is it working?
No. Could you please try again?
Oh, so let me now let
yeah,
me
yeah. Now
this is working.
Yeah, now we can see the X-ray crystallography slide.
Oh, okay, then I'll keep it like this. That's okay. Okay, and can you see the cursor now?
Yeah, we can see.
Perfect, I think that helps. So what happens when we solve the structure using X-ray crystallography? At the end of the processing that data, what you get is a beautiful electron density. Okay, now that electron density translates to particular structure. Okay, that is all governed by the parameters we use refining or building those structural models. For example, if we have proteins or biomolecules, or nucleotides, we have very refined parameters and constraints. So the structures of protein, like this part, we can resolve in atomic detail. But the big challenge comes when we have this fellow here, non-covalently bound drug molecule in the binding pocket. Now here we don't have many validated parameters which we can use for the direct refinement, and those structures are likely to be the less certain. And when we look at the protein data bank, which contains most of the structures which are deposited in public domain, as of this month we have close to 180,000 structures. Out of them, 90% come from X-ray crystallography, and out of them 35,000 structure at least contain one of these 25,000 small organic molecules which are of interest in drug design. So what happens when we look at this here, the electron density from crystallography? When you go from left to the right, the resolution kind of drops, but the number, of course, increases. And here we show an example of a tryptophan side chain. So when the resolution is very high, say for example 0.5 or zero point 0.6 angstrom, the density is clearly defined that we can fit an indole ring here. And as the resolution drops, we can see it becomes a blob of density, and we can put anything here which will explain that density. So most of the structures we have in the data bank fall in this range. And here, when it comes to the protein molecule, we can put the side chain properly into the density because we know the sequence of the amino acids when we crystallize the protein. But the big challenge comes with the drug-like molecule or the drug molecule in the binding pocket. So at very high resolution, where very few structures we have, we can clearly resolve the each and every atom of the drug molecule, and we know this is how it's going to bind in the binding pocket. And as the resolution drops, what happens? Your electron density becomes a blob. And the way you put a drug molecule can vary, and it's difficult to gauge whether it's going to bind this way, this way, this way, or this way. So what happens that when we have a very high resolution, your electron density dominates the structure, and we have more confidence in the structure. As soon as the resolution drops, the geometric restraints or the structure parameters we use for refining the structure will govern whether the structure is going to be good or not good. And I'll just show you one example from the literature here. Now here is a classic structure of a receptor called PPAR, a common target in diabetes and obesity a while ago. So one of the crystal structures was solved, and it was revealed that the structure is uh, crystallized in an activated state in the absence of any ligand. And they found there's a chain of water molecules in the binding pocket. And which was nice that they could get activated state like for an apoprotein with no ligand or no inhibitor. Now same group of researchers, after 2 years, what they did, they went back and reanalyzed all this data, and they realized that no, there is no, actually is not a chain of water molecule. There was a fatty acid in the crystal medium, and that is the one got crystallized here. And that is the reason the structure was not in the apo form, but it was bound to a ligand like a small fatty acid. And that is how it was crystallized in the activated state. And again, this gave them a lead that this is what could be happening. And this just shows an example of that, how you interpret this density has a very significant consequences or the impact on what you are going to see in the binding pocket. And one of the ways we use in computational modeling, that how do we validate that that structure is stable or this is what exactly is found to be. So one we can do is look at the energetics, that look at this crystalline protein and the ligand complex, measure the energy, but that becomes a static structure or the static model. What one can do is look at the time evolution of system using molecular dynamics simulations. So in molecular dynamics simulation, what you do is solve the Newton's equation of motion and look at the time evolution of system. That over a time period of time, the system will evolve into the more stable state. And then you use the classical mechanics. And here it just shows an example of a small dialanine molecule bound to vancomycin. And here the dynamics shows that how water molecules wiggle around and how the surrounding molecules moves around, and the dialanine has enough flexibility to bind in the pocket. And then here we can see that this complex is pretty much stable, and this is what is happening here. So when I started this work on looking at how the structures of the drug molecule bind to the protein, we wanted to develop some methods for free energy calculations. Now, to develop any computational method, what we need is a large data set where we know the answer. So what we did was look at this data set of glycogen phosphorylase. So it's a target for diabetes, and most of the inhibitors are analog of glucose molecule. And we have more than 50 high resolution structures and binding data in the publicly available protein data bank. Now this gives us an advantage that we have so much of non-structural binding data we can use to develop and test our methods, because here we know the answer. But the problem started when we started analyzing the structures. So here is an example of a structure of the same inhibitor published by the same research group over a period of time. So one structure, the blue region is called nitrogen and the red is an amide moiety here. And the only difference between these two structures is the orientation of this amide group in the ligand. But simultaneously, what happens if this amide is in this orientation, we have an asparagine in the binding pocket. Look at its side chain here and here. It gets flipped, okay? Which looks fine, okay? There's not, at first you see, all right, these are complementary to each other. But again, out of this two, one has to be correct and other has to be wrong. We don't know which one. And here we have complementary interaction, that this also forms hydrogen bond, this also forms hydrogen bond, but which we don't know whether both are possible or one is possible or one is favorable over the other. So what we did was that because binding of ligand and the protein is more like a thermodynamic property, we need to look at the binding free energy difference between these two states. So first things we did was, now take this molecule, look at this amide group and rotate around this bond and see how many states we can see using quantum mechanics. So what happens when we look at this molecule here, and this is the amide group here, we look at the rotation around this and generate the QM profile of this molecule, and we find there are three minimas, one here, second minima is here, and the third minima is here. Then we immediately go back to the crystal structure conformation. The crystal structure conformation actually is sitting as higher in the energy profile here. Then we went to the validation server at the time available called Hiccup and Valdig URL. We took the structure out of those web server and they were sitting further high in the energy. And what we realized that if we swap oxygen and nitrogen in these two structures, so if I swap oxygen and nitrogen, actually it gives me the QM minimum 2. And for this server, if I swap oxygen and nitrogen, it gives me the QM minimum 1. And there is an additional minima. So what is happening here? It looks very simple that you swap oxygen with the nitrogen, but then in the electron density it's difficult to identify whether it is an oxygen or nitrogen based on the resolution. So here just one swapping can lead you a structure with such a difference in the relative energy. But again, this is significant, but it's okay because we are looking at this ligand when it is in the free state in vacuum or in solution. The most important is out of all these 5 possible states, which is the states which is stable in the binding pocket of the protein. And remind you, the structure which is bound to the protein, that is our starting structure for drug design. So for example, if the structure is supposed to be this, okay, but the crystal structure contains this, and you start designing your drug molecules using that one, which is not correct, is most likely to fail in the subsequent steps in the drug discovery. So we need to be very sure about how we know exact how that drug is binding to the protein in the crystal structure. So we took all these possible states and calculate the free energy between those two states, all the states when it's bound to the protein. Now to calculate free energy, what we use is a method called thermodynamic integration. In this, what you do is calculate the work required for the reversible process from going from one state to the another state using a process called alchemic mutation. Now to simplify that, for example, we have a state A bound to the protein and there's a state A in the water. So the free energy of binding of A from water to the protein is delta G3. And for the state B, it is delta G2. Now delta G3 and delta G2 are the ones you measure by experiment. But for computational calculation, they are very, very expensive. But then we have a workaround. What we do, we take the molecule or the state A in the protein and mutate by alchemic mutation into the state B and calculate delta G1. And similar thing you do in the water. So based on the closed thermodynamic cycle, the difference between G3 and G2 is equal to the difference between G1 and G4, which we can calculate by computation. So we do this series of calculations to calculate the free energy between these two states when it is bound to the protein. Now this is what we find here. that this is a state which is bound in the crystal structure. We have QM minimum 1, QM minimum 2, and QM minimum 3. When we calculate the delta G between the states, for example, here when I go from this state to this state, you lose a 22.6 kilojoules per mole, which is telling me this is the most favored one with the numbers here. Similarly, this one between QM minimum 1 and QM minimum 2, you see minus 12.7 kilojoules, which is telling that between this and this 2, this is the one favored when it is bound to the protein. Now you realize when we were in the free state here, the lowest energy structure is this one. But when we are in the bound to the protein, the QM minimum is the lowest free energy state. So here we learn two things, that the conformation of the state of a given molecule, which is a global minimum in the free state, need not necessarily be the state it is bound to the protein. And second thing, we found that in the crystal structure, it was amide had a particular orientation. Actually, when we do the free energy, we realize actually swapping this oxygen and nitrogen, we get another state. This is a more stable state when it's bound to the protein. Now to look at the cost of the free energy between this state and what was in the crystal structure, it is about 23 kilojoules per mole, which is very significant, which is telling me that if I take this state for my free energy calculation or drug design, it's likely to fail. I need to use this state in the calculations. So at this stage, then we analyze some of the out of 50, we took another 5 or 6 crystal structures which had the similar groups like an amide group here, and see where they are sit on the profile. And not surprisingly, all these conformations which are in the crystal structure, they were either sitting here, 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 or here. Not a single conformation which was crystallized contained this state, which we found from the free energy calculation. This is a more stable state when it is bound to the protein. So this demonstrates that although we have so much high quality experimental data in the literature, it is always difficult to validate that whether the bound state we have is correct or not. So this is what we have at this stage. So before I make a small jump, so here the molecules were simple, like say glucose analogs. Now the big challenge comes when we do this free energy calculations or the molecular simulation, is that how do we describe these molecules, or what we call is the force field of the molecule. Now this force field parameters are very crucial when we want to refine the structure in X-ray or NMR, or run the molecular simulations or do the drug design. Now most of the biomolecules, including proteins, nucleic acids, lipids, and sugars, we have very well established parameters. Some of them include gromose, amber, jam, and OPLS. But when we have a small drug molecule or drug-like molecules, what we call heteromolecules, we need the force field of those molecules which could be compatible with the given biomolecular force field. And with the limitation of available parameters, what I had done, develop a semi-automated protocol where we can generate and validate these parameters. And then automation of that process led to something we call automated topology builder and repository. Now here just demonstrates what a force field means. That we have a molecule here with those atoms connected by bonds. The description of this molecule, which is split into two parts, bonded interactions and non-bonded interactions. And the bonded interactions combined of the stretching parameter for the bonds, bending parameters for the angles, and the rotation around the dihedrals. And some of the bonded parameters to retain the planarity. On the other hand, we have non-bonded interactions where we calculate the electrostatic interaction between the charged particles and what are the van der Waals interaction between various these particles. So combination of all this kind of gives what we call the description of the force field of the molecules. Now, most of the biomolecules we can generate those parameters very easily. But when it comes to the small drug-like molecules, it's a lot of effort. So we came up with this automated topology builder and repository about 10 years ago. In the right, it gives an outline of the blueprint of what is running in the background. That when you submit a molecule, It does various optimizations at QM level of theory, calculates the gets the optimized geometry, calculates the partial charges, then we combine that with the description of the biomolecules and combine those parameters to generate a description what we call the topology. Now, as of this week, we have about 15,000 users worldwide, and we have about 500,000 molecules in the repository for the small drug and drug-like molecules. Now, these molecules have been validated against what we call as a hydration-free energy. So here is given data about 300 molecules where we have the experimental delta G, and this is calculated using the ATP. And most of the molecules we have fall within the error bars of the experimental calculations. So now once we have this ability to generate description of any drug or any chemical molecule, now we can expand our study to the another data set and more data sets. So next data set we took was a protein called phenyl ethanolamine and methyltransferase. Now we have a collaborator at the University of Queensland who have been working on this target for many years, and they have generated a large number of binding and the structural data. So what we took, and particularly most of this work was done by the PhD student Pramod Nair. Now Pramod Nair, he did his B.Pharm from BCP and masters from NIPER, and then came over to UQ to do PhD with us. And what he did was take the series of the structures and run the molecular dynamic simulation to see if those complexes are stable. And the ones which were unstable, we then investigate why they are unstable. So here I'll just show two examples. That what happened is that they already had published a structure of rnor adrenaline, which is a substrate for this protein with this particular orientation and stereochemistry. Now, one of the structure which was unstable in the simulation was this fellow, which is a protein etabank 3-HCB. Now, this was an S-noradrenochrome, so and bound in this orientation, and this is an inhibitor. Now, there was a drastic difference between these two molecules. Now, this is a substrate with R stereochemistry. This is inhibitor with the S stereochemistry. And when we look at the orientation with respect to the glutamate in the binding pocket, they have exactly opposite binding mode. So what happened? This structure was published as a novel binding mode in the literature. Now, one thing we realized that when they were crystallizing this noradrenochrome with the protein, they did not just use the S isomer, they used a racemic mixture. And they took the electron density, and the electron density they interpreted as a S stereoisomer for this molecule and in this particular orientation. Now, this was one of the molecules when we take the system and run molecular simulation, it was unstable. So what do you mean unstable? So when you run the simulation, this molecule will just pop out of the binding pocket, telling that something is not right with this molecule or with the protein. So we went back and looked at the electron density in the binding pocket for this protein. And what we see here is that there are 4 possible ways we can put that molecule in the binding pocket. And then for each, for R isomer and for the S stereoisomer. So we have 8 possible ways this ligand could be placed in the binding pocket. So what was done was only one, this is the only way was published in the literature. So when we ran the simulation, out of 8, this one was unstable, this one was unstable. So one which was already in the crystal structure was already unstable. And other 2, this one, they were unstable. So this left us with the BS, BR, DR, and DS. So we have 4 possible states which were stable in the binding pocket. Now we had to find out of 4 which one is the most stable. Then again we ran the free energy calculations between 1, 2, 3 and the 4 states. So after completing the free energy calculations, what we found, DR, which is this one, is the most stable state in the binding pocket. So now we go back and look here. So this is what was originally published, that this molecule has the opposite binding mode and the opposite stereochemistry. But in reality, from the simulation, what we find actually it has a similar binding mode in the same stereochemistry. So this is again another example showing that what was originally published and what was actually stable in the binding pocket was exactly opposite. And any drug design effort using this structure would have led most likely to a failure. But then now we understand this is how it's supposed to bind in the binding pocket. Now to continue this example further for this protein system in PNMT, what they did, they use the fragment-based screening. What they do, they take a cocktail of 4 small fragments or drug-like molecules and then use to co-crystallize in a high-throughput crystallography experiment with the protein. So once they completed this study, they identified the 12 fragments and they published the 12 crystal structures. So we took those 12 crystal structures and run molecular dynamic simulations. Out of 12, 11 was stable and all happy, which is nice, but one was unstable. And then we were interested in understanding why that one is unstable, what is wrong there, something is not right there. So it's shown here. So that cocktail contained these 4 molecules. Okay, now this is the molecule here. Actually, they have put it in a crystal structure. Now this is a molecule was always unstable in the binding pocket. Then we realize, if I put this fragment or this fragment in the binding pocket, the density is too big for these molecules. And that won't explain why we see such a big density for the small molecules. When we tried to put this fragment, it was too big in the binding pocket and shown the clash. So what Pramod did was, when he put this molecule at one at a time for fragment 1 and fragment 2, they were finding binding in the different parts of the binding pocket. So what he did, he took both these fragments together and put it in the binding pocket and run the simulation. And it turned out that actually those 2 fragments are binding together. And this is why explaining the why this density is so big. And when we did the free energy calculation, what we realized that the combination of fragment 1 and fragment 2 had way better free energy than the individual fragments. So fragments are binding cooperatively in the binding pocket. So we took the experimental electron density and refined the structure using these 2 fragments. So now in protein data bank there are 2 entries. What they publish is this structure, and what we refine using simulation is this structure. So eventually, I haven't checked recently, I think this structure was removed, and now this is the only structure remains in the protein data bank, which explains that actually in the binding pocket we don't have this fragment, but we have these 2 fragments. And this gives a very good starting point to joining this fragment for the fragment-based drug design. But unfortunately, this work in the experimental group discontinued this project, so we could not go further building this molecule and testing it further. But again, this again shows another example that what is shown in the literature and what could be in the reality is totally different. And this also partly would explain this always difficult to design drug molecules based on just extra structures we know. So again, in the next slide, I'll just show you an overview of the examples where we have shown that what you see in the extracrystal structure and what we find in the reality may not always be the same. And one of the examples include the conformation of the molecule, the structure itself. It could be the relative orientation, how you place them, or it could be the stereochemistry S versus R, or it could be the tautomer or the protomeric form. So here I'll just add one more example of showing in for the tautomers or the protomers of the molecule. And here I take one of my favorite example of warfarin. So tautomers are molecular chameleons, and they can have multiple states or structure where they involves intramolecular movement of hydrogen atoms. Now the big question for drug design is that when the molecule can exist in more than one tautomeric or protomeric form, the basic question remains, is the tautomeric state same in the free state and the bound state or different? And again, that would govern whether you are likely to get success in your drug design efforts using that structure. So the example I'm going to use is drug molecule warfarin. Now, warfarin is widely used as an anticoagulant since 1953 and still in market. Now, there are few anticoagulants have been introduced in the last few years, but still at this currently, warfarin remains a drug of choice worldwide. And that its target is vitamin K epoxide reductase. Is only this year, or I think sometime last year, first ever structure of vitamin K epoxide reductase with warfarin has been published. And we only had structures of warfarin with the albumin or the cytochrome P450. Now it binds extensively to these proteins and has a very complex pharmacokinetics. If anyone in the audience have ever been on the warfarin therapy would know that warfarin has so many interactions that if you slightly change your diet or schedule or anything, your drug dose has to be changed for a given day. And then you do the prick and the blood clot time, and based on the blood clot time, the pathology or the doctor will prescribe the dose for that day or the next day. It's very severe, because if your warfarin level is low, you are likely to get blood clot, and if the level goes high, you're likely to get bleeding. So it has to be pretty much balanced. And the reason for that is that it is given as a racemic mixture. So here I'm just showing the R stereoisomer. Okay, it can exist in the neutral form and the zwitterionic form. But now this neutral form is most likely to be present in the acidic environment or in the lipidic environment. Now each neutral stereoisomer here can exist in the 20 tautomeric forms. So when you're taking a racemic warfarin, there are likely to be 40 plus to 42 chemical moieties floating around in your body, and which kind of indicates why warfarin has such a complex pharmacokinetics and monitoring issues. And again, these are difficult to study by experiments, because when you crystallize, you're going to crystallize one state. In the solution, there are more than one states present, and when you try to record the NMR, you have signals coming from all possible states. So it's difficult to deconvolute those signals and work out which are the most likely tautomeric state present in a given system. So here given are the 20 tautomeric forms of R warfarin, and similarly 20 forms will be there for the S warfarin. Here we have 4-hydroxyenol. So when you look up for warfarin in literature or PubChem or any database, this is the only chemical structure you will see, most likely. But then this enol can undergo tautomerism to form this enol. So this enol becomes keto, and this keto becomes enol here. Or you can have a diketo. But see, when you go from enol to the diketo, you introduce another chiral center, and then you have two stereoisomers. Similarly, on the side chain, there is a ketone group, which can also from enol and again introduce Z or the E stereoisomer, similarly for this one and the 4 here. And this can also exist in another enol form here, as it's shown. And the warfarin also has something called what we call the hemiketal or the cyclic tautomers. So what happens is a hydroxyl group here can attack the carbonyl carbon here and form the cyclic ether or the hemiketal. And when it forms a cyclic hemiketal, you introduce one more chiral center. So you got 2 diastereomers here and 2 diastereomers here, which gives this complex chemistry of this 20 tautomers. Now, what we want to do is try to understand out of this 20 tautomers, which are most likely tautomers, so we can understand how warfarin binds to its target. So what we did was look at the entire process computationally, that we have atoms at infinity. To form this molecule in vacuum, it needs something called what we call the heat or enthalpy of formation, which is a very fundamental quantum mechanical property. And then once the drug is formed, it is also composed of a component of entropy, which arises from the various rotation and vibrations of the molecule. And then measure the free energy required for this molecule to be transferred into a solvent, and which will give the solvation free energy component. So what we are trying to do is look at this entire process computationally and see if we can make any sense out of those 20 or 40 tautomers which are more likely to be present in a given environment. And again, for the formation of this process, we have used various combination of methods using direct method or homodesmic reactions method. And then we calculate the entropy using various methods using vibrational component and conformation of the molecule. And then you can calculate the free enthalpy or the free energy of solvation using implicit solvent or the explicit solvent. Now what I'm going to do, I'm going to make a small jump here and come here. So what we found actually when we did all these different components using various computational methods, out of those 20 tautomers, we found those that this first two, where we have keto enol and enol keto tautomer, and these two cyclic tautomers are more likely to be present in water. So out of these 20 tautomers, now we realize that these 4 tautomers are present within the 15 kilojoules per energy in the free state. Now we know by experiment that the binding free energy of warfarin with cytochrome P450 or albumin or its target vitamin K epoxide reductase is in the micromolar range, which is about 30 kilojoules per range. So for that kind of free energy of binding, the binding pocket can accommodate this, and this 4 tautomers for R warfarin and another for S warfarin. And again, here the electron density, we can't actually work out which of these are present or more than one of those are present. But at this stage, we understand that out of 40, 8 possible tautomers could bind to the protein, whether it could be the target or the albumin or the cytochrome P450. So the study allows us to narrow down from 40 to the 8 possible states. And out of those 8 possible states, now we can understand that most of the drug design efforts as of today or as of till now have always focused on this state, which is the open chain state. Now we understand this one of the cyclic forms or 2 of the cyclic forms are also likely to bind here. So which kind of gives an indication that maybe if we focus our effort on this form, we are more likely to get anticoagulant with the least side effects and maybe more potency rather than just focusing on this. So that gave us some indication on the direction of the drug design effort we should make for the next generation of anticoagulants. So here I just give a brief summary of what we just described till now. So what we have seen is that we can use a combination of molecular dynamics, quantum mechanics, and the free energy, of course with the force field parameters, so that we can understand how a drug beats conformation, orientation, protomeric state, tautomeric state, or stereoisomeric state, binds to its target. And again, these methods are meant to be go hand in hand with the experimental, say, X-ray and NMR approaches, and look at the complete picture. This is how the drug binds to the protein. Next, I will go to us briefly cover one of the aspects of what I'm doing nowadays. So as of now, what we look at the protein by itself when it binds to the drug molecule. So this is how example of given here is a SARS-CoV-2 COVID-19 spike protein. Now this is what we get from the crystallography, the protein. But in biology, or in reality, what happens? Each of this protein is decorated by these sugars on the surface of the protein here. Now, one of the key things which we have learned that in the crystallography or in experiments, generally those glycans are removed so that you can crystallize the protein. But again, this is not the complete structure. To look at the complete structure, we need to put those sugars back on that and look at the dynamics and see how those structures orient. And then we can use this information here to design potential drug or the vaccine molecules. So what I'm doing now is working on the methods where we can take this crystal structure from the X-ray, combine the glycan data which we can get from the various experiments including glycan array or mass spec or NMR or glycoinformatics, attach those sugars on this protein, develop those parameters in model, run the dynamics, and see how those sugars wiggle around the protein and how it affects the binding of drug and other proteins. Here it just gives a complexity of various sugars, conformations, and chemistry we need to take into account when we have to build this model. So here I just show you a small movie that we were trying to build the glycogen fragment or the glycogen molecule from scratch, which is made up of so many sugar molecules. And here we develop a protocol where we can evolve the system over a time from origin and open up into a sugar chains as per likely to be present in the biological system. And this is how the sugars would wiggle by itself. And what we are interested in is how the sugars wiggle around the protein and affect the binding of the protein. Now with that, I would like to thank you all for your kind attention. I would like to acknowledge a lot of people here. One of my previous mentor, Professor Alan Mark, and MD group, where I work at the University of Queensland. Now, these are the students who have contributed to the most of the work I presented today at the University of Queensland. And a lot of computing involved in this work has been supported by National Computing Infrastructure Australia and the QSAFE in Queensland. And also funding came from Australian Research Council on most of this work. And then we have current and former group members who have contributed to this work. Currently, I am at the Griffith University Institute of Glycomics, and again, mostly running computations using the national and the local facility. And Mark Warnitstein is my current mentor. And just to add, he is also the co-inventor of flu drug Relenza, which is in the market. And we work now on a range of antivirals design and discovery, including for COVID-19 and parainfluenza. And would like to end my talk with a small statement that, of course, the softwares and programs can do a lot of things, but they cannot replace the human intuition and the ability of error recognition of a scientist, and what I briefly call is like the scientific intuition, the chemical intuition of the scientist. So nothing is foolproof, neither extra structures, not experimental data, not the computational model, but it also had to talk to each other with the human intuition and or your ability as a scientist. Thank you very much.
Thank you so much, Dr. Alpesh. It was really amazing to listen to you and to know about your work in such a great depth. I know you tried to keep it simple, but though we had an amazing overview of your work. And yep, so yeah. So now is the time of the question answer session.
Sure.
So I would like Yash to read up some questions, but before that, we have our very first question claimed already by Dr. Elvis. So yeah.
Hi Alpesh, thank you for the wonderful talk. And this last slide, in fact, reminds me of sir telling us all the time that look at your results more than believing them first. So yeah, before Yash will go on flooding with questions. So there are a lot of students here who always wonder what is CAD like, what is a computational work like? How should we enter there? So could you address this and guide some students of how they can start studying this field?
Yes, again, most of this answer I'm going to answer based on my own experience. And see, when I started this work, to be honest, I never wanted to do computational work, not directly. So I was more interested in doing wet lab organic synthesis, peptide synthesis when I joined for masters. But for whatever reason, I ended up with a supervisor who was doing computational work. Then I said, this is fine. So there are multiple ways. Now, the resources we had at the time, I'm talking about 20 years ago, were very limited. Now we have internet is flooded with the resources. If you want to get into this field, it's a very diverse and big field. And again, what I'm doing in the computational work is just a small fraction of a big computational field. So if you want to enter here, just start somewhere. You are better off to starting with a small project with a supervisor like say Professor Coutinho or others in the field, or his students who are already establishing their own things, and working with them on a small project, and they will guide you to do certain things, and that's how you start. So this is how I started, and out of interest I started expanding into more and more and more areas. So when I was doing my PhD, of course we were doing NMR work, but sir also had more interest in developing QSAR method, quantitative structure activity relationship, that you have a series of chemical structures and you have a series of biological data. Now you want to determine some properties of those molecules and see if you find a correlation between those properties and the data. If you can find a property a relationship, then we can design next molecule which would have improved binding or the improved biological activity. That was a simple concept. And we started working on various alternate methods. So again, my advice here would be start with a small project or a sub project of your masters or PhD in this area with the right supervisor, and then go from there.
Thank you, Alpesh. So yes, Yash, you can take ahead with more specific questions from the participants.
Yes, sir. So the first question is, before the target validation step, we need to know the mechanism of how a particular microorganism affects our body and which receptor does it bind to. So how is that determined?
So again, what I'm presenting today, it comes at the stage we have identified the drug binds to a particular target. Now, so for microorganism or antibacterial, antiviral, a lot of work has been currently going on where they take the drug molecules, add to the culture of the virus or the bacteria, and they find inhibition or the what they call EC50. Now, based on the uh, some of the markers in your experiments and the nature of the drug molecule, you narrow down what are the likely targets present in the microorganism. The next set of experiments will involve taking those one target at a time or in parallel and test those molecules whether they bind to that particular target. Now that becomes very, very challenging for antimicrobials in particular, where some of the drug molecules might act using more than one mechanism. Either they disrupt the lipid bilayer or the bacterial wall, or they're binding to its DNA or doing something else, or maybe they are just preventing their entry or sticking to the cells. So there are multiple mechanisms. But most of those narrow down of mechanism will be based on existing knowledge and trial and error.
So the next question is, in the activation of tumor pyruvate kinase M2 using synthetic modulator, what assistance may be provided by Glycomics?
So, okay, now glycomics is a very diverse field, as I said, that most of the proteins in the body or microorganisms, especially the tumor cells, glycans play a very, very, very critical role. Now, some of the work which has been done at the institute where I work, they are looking at the glycan array and analyzing those glycans using mass spectrometry. And what they are able to pinpoint is that the glycan structures or the types of glycans you find on the protein are slightly different or significantly different in the tumor cells compared to the normal cells. Okay, now this is general statement. Now for a specific target, we will be able to find specific things, and which could be used as a biomarker to identify particular type of tumors, which is work in progress at many research groups. So to come back to your specific question, that of course glycan will play a crucial role in the protein you mentioned, and it would be worth investigating the uh, glycan nature or the glycan structure for your protein. Yeah.
Thank you, sir. And one final question is that before MM2, we do energy minimization in ChemDraw, but when we deal with biological system parameters of drug, when we deal with biological system parameters of drug changes up to a certain extent, how will we manage this free energy of the drug?
Again, what you are using ChemDraw and other packages, it's limited in its ability to do certain things. Okay, so if you're using ChemDraw for Office for drug molecule, I don't know what kind of force field or parameters it is using. Uh, if you're looking for a particular property, you are better off using right program and the right software and the right force field. That's what I would suggest. So as I said, again, using a particular program, say ChemDraw, could be good for a minimum, structure minimization or looking at those, structures in 3D. But if you want to calculate certain properties, then those structures have to be optimized using certain level of theory or certain force field for accurate estimation of those parameters. So again, if you want to do a series of molecules of interest, you're better off working with someone who has done similar computational work as a mentor.
Thank you, sir. Now Keitki will take over the questions from the YouTube comments.
Thank you.
Yeah, hello everyone. So we just have a couple of questions from our YouTube as well. So I'll start asking. So Aditi asked, what is the correlation between covalent and non-covalent bonds and crystallography? It was mentioned that structure of non-covalently linked ligand molecules are less certain by X-ray crystallography. If there is a presence of non-covalent bond, it would be an ionic bond, which gives a good structure, good crystal structure.
Okay. Yeah. Sorry. Before I go further on that comment, so just to clarify, when we say non-covalently bound ligand, it means there is no covalent bond between the ligand and the protein. Okay, so the only thing is the non-bonded interaction, which could be ionic, hydrogen bond, van der Waals, is a combination of everything.
Okay.
So it's not just a single bond between them. It's showing that those molecules are sitting next to each other and they're interacting non-covalently, means they're not bound to each other with the link. If it's bound with the link, it's easy to refine. Then we know where the link is and how the molecule could be placed. Since there's no link, it can tumble and go anywhere.
Okay.
And then we need to measure which is the correct way it is sitting in the binding pocket.
Okay, thank you so much, sir. So that's what her question was, that why is that structure is less certain. So that was her main question.
Yes, and again, why that is less certain? That's the exact thing. So if I have a protein here and I have a ligand here, if there's a covalent bond between them, the only way is going to bind. We know the location of the atom where it's going to bind, and then we can place the ligand correctly. If there's no bond here, now this molecule can rotate and move around anywhere. And that's where the uncertainty comes from. That's why we say it's less certain.
Okay, thank you so much, sir. And Aditi, I hope that answers your question. So the next question is from Chirag Gala. So he asks, do we also test warfarin effect based on INR ratio, apart from their binding domains to receptor and all those physiochemical characters?
Sorry, can you repeat that?
Yes, sure. So he asked, do we also test warfarin effects based on INR ratio, apart from their binding domains to receptor and all those physiochemical properties?
And again, if I go back to the warfarin, so the INR ratio, the measure that you are getting. So one of the things you are getting in the reading is a combination of what I described today. So we don't know which tautomer is binding to the target vitamin K epoxidase. We don't know which tautomer is binding to the albumin or cytochrome P450, or which is a component present in the blood. Or it could be a combination. That's the reason we get so much of fluctuation with the slight change in the diet or thing, and the INR ratio will fluctuate, and you have to change the dose. So of course, yes, we do have to take into account all possible states and all those physical chemical properties, but at this stage we are still not there yet that we can segregate and identify those tautomers.
Thank you so much, sir. Hopefully that answers Chirag's question. So over to you, Sakshi now.
Yeah, thank you so much, sir, for answering all the questions very nicely. And I hope that we all have our concepts cleared by now. So now passing over to Manali for a formal vote of thanks that we have as our tradition. So yeah.
Thank you,
Manali.
Sakshi. Yes, thank you, Sakshi. Good evening, everyone. I am Manali Somana, Vice President of American Chemical Society, Bombay College of Pharmacy Student Chapter. And it is my honor and pleasure to give the vote of thanks today. Truly, it was a very engaging webinar on drug binding at an atomic level. I would like to thank our speaker for today, Dr. Alpesh. Thank you for sharing your knowledge and expertise on drug binding in such a great depth with us and for addressing everyone's doubts with ease. Thank you for taking time off your busy schedule to come here today. I would like to thank our faculty advisors, Dr. Krishna Iyer, our co-advisors, Dr. Sagar Patel and Dr. Elvis Martis, and the whole ACS-BCP student chapter team for organizing and managing today's webinar. I would also like to thank the undergraduate, postgraduate, PhD students, faculty members who have attended this event and have engaged in it with such great interest. We hope you all had a great time and an informative evening with us. We have posted the link for the feedback form in the chat box. Please fill it as soon as possible. Lastly, I would like to request everyone to switch on the videos for us to take a screenshot of today's participants.
Thank you everyone for attending. So I think whenever I had a chance to visit Bombay, especially BCP, so I think it was like 2 years ago, whenever I go BCP, of course, meet sir and we'll have a small chat or the seminar with students. So this is first time it's happening virtually. Mostly it happens when I'm at BCP. And it's interesting, when I started my B Farm in 1996, so it was 1999 when Krishna sir joined BCP. It's the first year. And got interesting memories with sir, and Coutinho sir used to teach us chemistry, still teaches chemistry, and everyone's favorite. And Krishna sir and yeah, we used to travel together on the same local, same bus.
By the way, Alpesh, Krishna sir is now the in-charge principal of Bombay College.
Oh, that's a good news.
Yeah.
No, lot of developments, eh? That's nice.
Just requesting.
Did you take the screenshot?
Yeah, I just want to request if you could stop your screen sharing so that we can have a full frame of people.
Sorry.
No, not a problem. Yeah.
So is it working now?
Yeah. Yeah, so when we are done, please let us know.
So all good. Let me know when time to switch off.
Yeah.
It's done. Thank you.
Okay. Thank you, everyone.
Thank you, everyone.
Thank
Thank
you,
you, Dr.
sir, and
Alpesh.
thank you,
This was really
everyone.
great.
Yeah, best wishes. Thank you.
Thanks, Alpesh. Good night to you.
Thanks, Elvis. Finally.
Yes, thank you so much.
No problem. No problem. We'll catch up again on our other group anyway. So take
Yes,
care.
definitely.
Yeah.
Yes, bye. Good
Okay.
night.
Thanks. Bye. Bye.